Hello, welcome to Science Breakthroughs from Science Mission. I'm Sadashiva Pai and going to discuss a couple of breakthrough papers published in the week of July 21st. Let's start. First, we will discuss a paper published by Ruta Ishimura in Science on the role of t-RNA in neurodegeneration. Why is this paper so important? Role of translational machinery in neurodegenerative diseases is not known. Authors demonstrate a point mutation in tRNA NTR20 leads to cerebellar ataxia. Introducing NTR20 to mouse with the disease blocks cell death. Let's look at the data. Authors observed progressive apoptosis of neurons in the inner granular layer of NFF205 mutant cerebellum in week 3, and the animals died in 8 to 9 weeks of age with ataxia. Apoptosis was also observed in dentate gyrus, CA2 pyramidal neurons, layer 4 cortical neurons, and retinal photoreceptors. Western blot analysis of mutant cerebellar extract failed to detect protein GTP-BP2 as authors identified the NFF205 mutation as a point mutation in the intron 6 of GTP-BP2 gene that results in misplaced mRNAs with premature stop codons. Pelota was immunoprecipitated by GTP-BP2, and GST-GTP-BP2 fusion also pulled down pelota from brain extracts, which demonstrated that GTP-BP2 can interact with endogenous pelota. Authors identified a mutation T2C in NTR-T20 gene of B6J strain at the nucleotide 50 of tRNA T-stem, which when processed turned into 105 nucleotide product rather than normal 115 nucleotides as shown in this figure. When wild type NTR20 was expressed in NFF negative mouse, the cerebellar and retinal death was rescued after 6 months as shown, thereby confirming the role of tRNA in neurodegeneration. Expression of NTR20 and its human ortholog were confined to CNS. Its level fell 50% at P0 to 30% at P30, with the concomitant increase in unprocessed tRNAs. Also, BJ6 strain had higher level of unprocessed tRNA in cerebellum than cortex and hippocampus. Authors also found that number of AGA pauses increased dramatically in B6J NMF350 negative cerebellum. Unusual AGA pausing was not observed in cerebellar libraries with NTR20 compared to B6J and B6J NMF205 deleted cerebella, indicating ribosome pausing during translation occurs specifically at AGA codons. So authors identify a translation mechanism for neurodegeneration. Next, let's look into a paper published by Chiara Manzini and colleagues in Cell Reports. Authors demonstrate that control of human intellectual and social functions by CC2D1A. Why is this paper so important? Genetic variants of autism spectrum disorder and intellectual disability with some identical physical traits are not clearly understood. Authors identify coil and C2 domain containing 1A, that is CC2D1A gene mutations in families with diverse intellectual and social disabilities. Authors demonstrate that reduced dendritic complexities in CC2D1A knockout neurons and were rescued with NF-kappa B inhibitors. Authors found 4 families with a total of 16 individuals affected by spectrum of cognitive and social impairments including ASD, NSID, and seizure. Genomic analysis and further sequencing revealed G2T mutation in 3 of 4 families and a deletion in the 4th family on CC2D1A gene. Complete exclusion of exon 6 is seen in family 1, when analyzed for CC2D1A mRNA. Western blot analysis of patient cell lines showed reduced CC2D1A expression in heterozygous carrier and complete absence of CC2D1A in the patient, either in the full length or truncated form, showing that this mutation is null. A simplified dendritic arbor was observed in cortical neurons after electroporation of shRNA at P11. Reduced dendritic complexity was also evident in CC2D1A knockout hippocampal neurons, and dendritic spine density was significantly reduced, but synaptic density remained unchanged. CC2D1A overexpression caused significant activation of NF-kappa B than other pathways, while CC2D1B overexpression did not elicit an increase in NF-kappa-B activation, indicating that NF-kappa-B activation is specific to CC2D1A. No effect of CC2D1A depletion of NF-kappa-B at early differentiation, but NF-kappa-B activation was moderately and significantly increased above control conditions during the peak dendrite extension at 7 division in CC2D1A knockout neurons. Treatment with SN50 peptide inhibitor and JSH23 small molecule inhibitor but not inactive control peptide SN50M rescued dendritic complexity in CC2D1A RNA treated cells. This study demonstrates a role for CC2D1A in human intellectual and social ability. Thank you for watching Science Breakthroughs from Science Mission.